let's pull back the curtain and dive into this incredible, hidden world that all starts with a simple, familiar moment you've probably experienced yourself.
So you've had your blood drawn, right? But have you ever really stopped to wonder what happens next? You know, it's so much more than just a little tube disappearing behind a door. It's actually the beginning of a super high-stakes journey.
I mean, this is a voyage where that tiny sample gets transformed into a potentially life-saving diagnosis. And believe it or not, it all kicks off with just one person. So we're starting right at the beginning, at what is probably the most crucial moment of all, the collection itself. This is where the entire foundation for an accurate result is laid. Yeah, this isn't just some routine procedure. You should think of it like the launch sequence for your sample's entire mission. And the phlebotomist, they're basically the mission controller. And what they do in those first few minutes can literally make or break the whole thing. Okay, so let's get into what makes a perfect collection. There are basically 3 non-negotiable rules here. These are designed to protect the sample, and more importantly, to protect you, the patient. All right, these aren't just suggestions, they are absolute must-follow safety rules. Rule number 1, you gotta verify 2 unique identifiers. We're talking comparing what the patient says with their armband, no exceptions. Rule 2, the tubes have to be labeled right there, at the bedside, not before, not after you've walked away. And rule 3, and this one's easy to forget but so important, those tubes get inverted like 8 to 10 times right away. This mixes the blood with the additives inside and stops tiny little clots from forming. And here's why that's so critical. This quote says it all. A failure to label can result in wrong blood in tube and catastrophic incompatibility. So wrong blood in tube stands for, well, wrong blood in tube. And, it's exactly as scary as it sounds. A simple labeling mistake isn't just a minor oopsie. It can mean a patient gets the wrong blood type. And that is, without a doubt, a catastrophic, potentially fatal mistake. And the precision doesn't stop there. Check this out, the order of draw. It looks complicated, but it's pure genius. See, each of those different colored tops has a different additive inside. Drawing them in this exact order prevents any of those additives from cross-contaminating the next tube. Because if that happened, it would totally throw off the results. It's all about keeping each sample pure. All right, so the sample's been collected perfectly. Now it's time to follow it into the hidden world of the laboratory and see where it goes. Now, you might picture the lab as, one giant machine, but that's not it at all. It's really a series of super specialized departments. Think of your little sample tube as a messenger arriving in a huge city. It has to find the exact right district to deliver its crucial message. And one of those districts is serology. And this is where the real detective work happens. It's just amazing how one sample can be interrogated to find all sorts of clues. We're talking invaders like syphilis, signs that the body is attacking itself in an autoimmune disease, or even confirming the very beginning of a new life. It's all about finding the antibody clues our immune system leaves behind. But of all the destinations in the lab, one of the most critical has to be the transfusion service. You probably know it as the blood bank. And in here, the stakes are just unbelievably high. The whole goal is to make absolutely sure that any blood a patient receives is a perfect, safe match. And they do that with this powerful 3-step safety check. It's a type, a screen for any unexpected troublemakers, and then a final cross-match, which is basically a dress rehearsal in a test tube. It's all about preventing a dangerous reaction before that bag of blood ever leaves the lab. Okay, so we've covered antibodies and blood types. But what about the really complex questions? For that, we have to go even deeper, right down to our cells and our genes. And here are 2 incredible ways to do that. On one hand, you've got cytogenetics. Think of this as looking at the body's genetic blueprint, the actual chromosomes. Scientists are looking for big structural problems, like a whole chunk missing or an extra copy. Then, on the other hand, you've got flow cytometry. This is more like a high-speed head count. It's like facial recognition for millions of individual cells, identifying them by the unique markers on their surface. It's unbelievably fast and essential for classifying diseases like leukemia. Now, if we're looking at a piece of tissue, not blood, we turn to histology. This is all about the tissue's architecture. It gives doctors a view of the whole neighborhood to see how all the different cells are organized and living together. Is everything neat and tidy, or is there a tumor disrupting the community?
And then there's cytology. If histology is the neighborhood, cytology is about checking out the individual residents. This is where they look at individual cells that have been scraped or collected, looking for any suspicious characters that might signal disease, like in a pap test.
So, wow, after all this incredible analysis, how does all this data become a diagnosis? How does it get back to the doctor and actually start making a difference?
Well, first, you got to understand that not all labs are created equal. You have satellite labs, which are all about speed. They're right there in the action, in the ER or the NICU, giving doctors urgent results in minutes. Then you have reference labs. Their focus is on scale and specialization. These are huge off-site facilities that handle massive volumes and run all the super complex and rare tests that might take days to complete.
But here's the thing. Whether it's a satellite lab or a reference lab, every single result is a critical piece of the puzzle. The entire journey, from the patient's bedside to the lab bench, is all about one thing: turning a physical sample into actionable, life-changing information for the doctor. And it's just amazing when you think about it. It all boils down to this: one single, tiny sample, collected with so much care at the start of its journey and holding the potential to provide hundreds of life-saving answers, guiding everything from a complex diagnosis to a safe blood transfusion. So the next time you see a blood sample being taken, maybe you'll think about it a little differently. You'll think about that invisible, high-stakes journey it's about to begin. It's really a testament to the science and to all the hidden heroes working behind the scenes to keep us safe.